Thank you. I guess I'm the older generation. But when I began here, I was the younger generation. Okay. Okay, so like Michal said, I'll be speaking about using our research over the last 35 years, which is basically built on using wild species of tomato as a source for improving food quality. And just to put things into perspective, most of our research revolves around sugar metabolism and sugar content, because sugar content determines fruit quality, even in a fruit like tomato, which you don't think of as sweet. When we think of sweet fruits, we think of melons, sugar cane, sugar beet, grapes. We don't think of tomatoes, but sugar content is a goal in all of agricultural research, but even in tomato. And since a number of people have already stressed how Volcani Center is a center that's based on a mix of basic and applied research, our approach was to see if we can understand the metabolism of sugar and carbohydrates in the developing tomato fruit, so that we can then try to manipulate or change or improve certain bottleneck steps in the metabolic pathway, under the idea that the tomato fruit, as it develops, is receiving sucrose from photosynthesis. Question is, what does that tomato now do with all of that sucrose? So basically, all cultivated tomato fruits follow this pattern of sugar accumulation, where in the beginning there's a small amount of sucrose, but at the end there's no sucrose that accumulates, disaccharide sucrose, and you get approximately equal molar amounts of fructose and glucose, two hexoses or two monosaccharides that comprise sucrose. But if you think about it, most high sugar fruits, sugar cane, most of the fruits that we know, melons, they get to very high sugar contents and sweetness because they accumulate specifically sucrose. So how do they do that? And why don't tomatoes do that? Now, it turns out that there's no genetic variability for sucrose accumulation in the cultivated tomato germplasm. It doesn't exist because of the evolutionary history or the domestication history of tomato. Tomato, as you know, began in South and Central America, and after the discovery of the New World, moved over to Europe, so that all of the cultivated tomato varieties that we're familiar with are tomato varieties that went through a certain genetic bottleneck that Roy Ben David mentioned earlier, and then came back to the New World and expanded throughout the Old World, but it's a very, very limited germplasm. So what we had to do is to go out of the cultivated species and to utilize natural genetic variability to improve food quality. And this is a collection of some of the wild species of tomato relatives. This one we use a lot, the Habrucheates. It's a very hairy tomato. But then there's also the Pinellii and Schmilievskii. And in fact, we even have one 3-species hybrid that I'll speak about in a minute. Okay. Now, the use of wild species for introgressing and breeding, improving tomatoes has been known almost 100 years already. It's been used for about 100 years. And in the beginning, obviously, it was used for things such as pest resistance, disease resistance, because those are attributes that you associate with wild species. They do better in the wild. And this is the first paper that came out on transferring leaf mold resistance from the closely related but still wild species, Solanum pimpinellifolium. The first report, and the first one who had the idea to use wild species, which I forgot to mention are usually either poisonous or they're certainly very bad taste, to use them as a source for improving taste was Charles Rick. And Charles Rick in 1974, so we're about 50 years ago, put out what was called the Rick's high sugar line, and that was based on a introgression from the wild species that's now called Schmilievskii. Then it was called minutum. And nobody knew, and to this day nobody has shown conclusively what is responsible in the minutum genome that increased the sugar content. It turns out we did a RNA-seq analysis of the line, and it has not just one single introgression, it actually has 11 different Shmilievsky introgressions in it, one of which is a gene that I'll speak about a little bit later, Lin5. And this is most probably the cause of the high sugar content in the Rick's high sugar line from Shmilievsky, but I'll come to that soon. I'll skip that. Okay, so in order to utilize natural genetic variability, we wanted to do it in a method that's directed and scientific and that makes sense, and not just to cross and to pray that we'll get something good. So we established a database of gene expression of all the wild species fruit through development, so that from over 100 different RNA libraries, we have over 2 trillion reads, and we now have a database that we're now making public of all of gene expression, fruit gene expression, 35,000 genes in the tomato genome, throughout all of fruit development in all of the wild species and the cultivated species of tomato. And this is going to be, I think, a very, very exciting and very useful database, because any researcher now can go and look for his favorite gene that he's interested in playing with. He can see how it develops, the expression of it develops throughout fruit development in all the wild species. He can then look for genetic variation in expression in the ripe stage through an even larger number of accessions. And since it's RNA-seq based on sequencing, he can even identify sequence polymorphisms, which, if he's familiar with a particular enzyme, could be very, very useful in determining the activity of the enzyme. So this becomes now an intelligent database that can be mined looking for a specific parent that can be used in a breeding program, and that would actually come with a molecular marker already in the information. So I'm going to tell you how we used a lot of this information, a mixture of our biochemistry studies and genetic studies to improve a number of different traits in tomato. The first one I'll talk about, because this is the one we've been working on the longest, is what I began with, sucrose versus hexose accumulation. If we can somehow or other try to convince a tomato to accumulate sucrose instead of hexose, maybe we can get a high sugar tomato. So it all begins with the following story of invertase activity. Invertase is the enzyme that hydrolyzes, that breaks down the sucrose that's coming from the leaf into the tomato fruit. And you can see that, as I showed before, tomato fruit themselves, cultivated tomato fruits, don't accumulate sucrose, because all the sucrose that comes in is immediately hydrolyzed by the invertase enzyme. Whereas all the green fruited species, most of the wild species that you saw before, here, for example, Habrochaetes, you see it does accumulate sucrose. It has actually a very high sucrose accumulation level, and that's because it loses its invertase activity through development. So that sucrose that comes into the fruit can accumulate. And that's according to this model where sucrose comes in. It can be hydrolyzed in the cell wall. We're not going to talk about that now. We'll come back to that later. But otherwise, it would go into the cell, into the vacuole, and the invertase will break it down. And you get a tremendous increase in invertase gene expression and in protein level in the vacuole in the cultivated tomato fruit. But in the wild species, you actually get a cessation. You get a low invertase expression in the young fruit and a cessation of invertase expression in the mature fruit. So we developed sucrose accumulating tomato lines that have the Habrochaetes allele for invertase. And in fact, they do accumulate more sucrose, but unfortunately, they didn't accumulate more sugar. They accumulate sucrose in place of the hexose. And we were very bothered by that because we thought it was a good idea. So we then went to the next step. We crossed that material with a third species, with Shmilievskii, which also has high sucrose levels. And in those segregating populations, we got what 1/4 of what I just described before of low sucrose accumulation, but we got 3/4 of very high sucrose accumulation. And when we then mapped that trait This is work from Daphne Marone and Marina Petraikov largely. We identified that the allele that we transferred from the Shmilievskii is an allele for a vacuolar invertase inhibitor. So it's an inhibitor protein that inhibits the activity of invertase. And if these are the two molecules, if this is the invertase enzyme and this is the proteinaceous inhibitor, the proteinaceous inhibitor completely knocks out invertase activity, because it turns out that invertase activity, even if it was expressed to a low level, is maintained throughout fruit development because it's a very, very stable enzyme, has very, very low turnover. But when we introduce the Shmilievskii inhibitor gene, and here you can see, taken from our database, these are the Shmilievskii accessions, the level of expression of invertase inhibitor gene, very, very high levels. And in our backcross lines, we now have lines that have 30-fold invertase inhibitor at the later stage of development. Now we can develop, finally, tomato varieties that, for example, these are all segregating F2 and F3 plants, each point of glucose, fructose, and sucrose levels. So with the invertase from Habrochaetes and the inhibitor from Esculentum in the Esculentum background, we get some sucrose accumulation, but not enough to actually increase the total sugar level. But when we add the Shmilievskii species invertase inhibitor protein, then we get a very high sucrose accumulation level with a net increase in total sugar levels. So these sucrose modifier lines are being, hopefully in the next couple of years, the first varieties of them will be coming out with the breeding companies that we work with. And they are really extremely tasteful. And they can increase a significant amount of sugar accumulation. Now, I mentioned, this I'll just do as an aside, I mentioned that the Esculentum, the cultivated tomato, has a tremendous increase in invertase expression. And in fact, it's the most highly expressed gene in the tomato fruit. And it's expressed to levels that are way above anything that's physiological. And the question is why? So this is doctoral work from Michal Moy, where we found that in all the sucrose accumulating species, that invertase activity is lost through development. There's a 166 base pair sequence in the promoter region of the enzyme. What happened in all of the other species, which invertase expression is increased, there was a duplication of that 166 base pair region, anywhere from 3 to 7 or 8 copies. And it's that duplication of the repeat units that is responsible for the expression, the super high expression in the ripening fruit. And here we added GFP to different constructs of either 1, 2, 3, 4, or 5 repeat units with GFP. And you can see that with the increasing number of repeat units, you get increasing GFP expression. And it increases from development from breaker to red fruit, just like we expect. In other words, there are compounds, there are transcription factors, as we'll see, in the developing tomato fruit. And not only the developing tomato fruit, they're actually in the developing wild species tomato fruit also. And that's why wild species tomatoes also ripen. These are the ripening responsible transcription factors. And what happens here is that the duplication, this R repeat unit, has turned invertase into a gene whose promoter is now sensitive to those ripening transcription factors. And you can see that here. This is the 166 base pair sequence of different backgrounds and different lines, the green, the first 3, the last 4. And you can see there are a number of different polymorphisms. 2 of these polymorphisms are in transcription factor binding sites. I'll show it here. And here you can see, and we've shown now by luciferase studies, and we're trying to do it now with certain transgenic plants also, that there are now binding sites for RIN that weren't there before. There are binding sites for FUL1, which aren't, and those are the 2 major tomato fruit ripening transcription factors. and also for one of the GRAS transcription factors. And in fact, what we've done already is we've overexpressed these transcription factors in tomato fruit under 35S, so that the transcription factors aren't ripening related, that they normally are, but in fact they're already expressed in the green fruit, in the immature fruit. And you can see that the control, which is just the 4 repeat units and GFP, follows this type of GFP as the normal transcription factors are expressed. But if we have 35S overexpression of the transcription factors, we have very high GFP already at the immature green stage. So in other words, this duplication and making available to the invertase gene a promoter region that's now sensitive and activated by the major fruit ripening transcription factors has turned invertase, even though it's not a ripening gene, it's hijacked onto the ripening gene family. So in other words, there probably wasn't any selective advantage to this evolutionarily. It's a hijack that just fortuitously turned into sucrose, turned into hexose accumulation. All right, so that's the story of sucrose. And more or less, we're basically finished with that story, and there are now prototype varieties, and we hope in the next couple of years some very good, tasty varieties will come out of it. So we looked at the invertase and the vacuole. Now we're going to look at, can we modify starch synthesis? And this was the doctoral work of Marina Petrikov, who studied the sucrose to starch metabolic pathway in tomatoes. And you don't think of tomatoes as a starch accumulating tissue, but immature green and mature green tomato fruit have a tremendous amount of starch. When I say tremendous, it's not really so big, because it's a high concentration of starch, but only when the fruit is very, very small. So the total contribution to the carbohydrate pool to the tomato fruit when it's ripe is going to be very, very small. What Marina found was that one particular enzyme is the bottleneck enzyme here. It's ADP glucose pyrophosphorylase. And when we look at gene expression of all the wild species, we see that one of the major genes that is different between the hexose accumulating and the sucrose accumulating tomatoes is the level of expression of one of the subunits of ADP glucose pyrophosphorylase, the large subunit 1. The enzyme is a heterotetramer. And when we transfer, or when we introgress the wild species large subunit 1 allele from Habrochaetes into tomato, this is what we get. These are tomato fruits, isogenic lines of tomato fruits, 10, 15, 20, 30, 40, 50 days. And this is a Western blot of the large subunit and a Western blot of the small subunit, the 2 major subunits of the enzyme, different antibodies, obviously. And what you see is that with Habrochaetes, by 15 days, expression of the large subunit is higher, and it preserves the expression of the small subunit, or the protein of the small subunit, because the complex is stable, whereas the monomers are unstable. And by the time you get to 20 days, we've almost lost the esculentum alleles. And by the time you get to 30 days, there is no more esculentum ADP glucose pyrophosphorylase, but the Habrochaetes alleles are still working. 30 days is when the fruit is almost full size. So you're getting the following. You're getting now, if we look at a per fruit, not per gram, but per fruit, this is the reservoir, we see that the starch level in the 30-day fruit is twice as much, and that starch is going to be broken down, like it always is, to sugar, and we see that the sugar accumulates in place of starch. So that we have now turned a tomato whose starch reservoir didn't contribute to the soluble sugar content to a tomato that starch reservoir can contribute, and contribute significantly, could raise the Brix levels by about 1. And this gene, together with the next gene, are also in prototype stages with breeding companies that we're working with. What's interesting is, and this was completely serendipitous that they found it, when Sanwan Wang went through 400 or 500 different accessions of tomatoes and did resequencing, he found that a number of the tomato processing lines that he studied, there was actually an integration from Habrochaetes for the large subunit of the ADP glucose pyrophosphorylase. In other words, for some of the breeding companies used the habrochaetes to increase the sugar content of the tomato. Okay, another strategy that we took is, if you remember, in the cultivated tomato, there are equal molar amounts of fructose and glucose. Fructose is twice as sweet as glucose. Sucrose is an average of fructose and glucose. So if we can make a tomato that accumulates mainly fructose instead of glucose, we could have, for the same amount of calories, same amount of sugar, we could have a sweeter tasting tomato. What we noticed is that the wild species of tomato, in addition to accumulating sucrose and having very low hexose levels, but those hexose levels are in a ratio of 3 to 1, 4 to 1, 5 to 1 fructose to glucose. So we were wondering if that is an independent trait, and in fact it was an independent trait, and this is work of Arik, Shammai, and Marina, on the identification of this gene. We called it the FGR gene, fructose-glucose ratio. This is work that was done together with Ilan Levine. And the gene was mapped and identified and characterized as a sweet transporter. It was then a new family of sugar transporters in plants, especially in tomatoes. And what it does, it actually extrudes, it effluxes glucose. It's a glucose transporter, but it effluxes glucose from the vacuole so that more fructose can enter. And by that way, it increases the fructose to glucose ratio. But just to give you an indication of what the wild species can offer us, so this is the gene. We can see it's only expressed in wild species, and it's not expressed in any of the tomato varieties. But there are a number of other sweet transporters that are also only expressed in wild tomatoes, and each of these then becomes a very, very exciting trait. And this material already is in, I think, a very successful cultivated variety in Europe. It's called the Nebula, and it has a high fructose to glucose ratio and a very special sweet flavor, and it really is very, very tasty. Next generation varieties are coming out of this, which will be more horticulturally, I think, successful. Okay, and the final trait I'd like to talk about, I have 5 minutes still? Okay. The final trait I'd like to talk about is the acidity of fruit. Until now, I've only spoken about sugar. But there are three major components to what we think of taste of tomato. One is the sugar level, one is the organic acid level, the pH, the acidity, and the third is the volatiles. Now, you can have good volatiles and you can have good acid. If you don't have good sugar, you won't have a tasty tomato. So sugar is the number one trait. Volatiles and acids come afterwards. And here we can see that most fruit, practically all fruit, are basically somewhat acidic. Some obviously more acidic than others. Tomato is somewhere in the area of 4, 4, 5. Melon is the most unusual fruit, that it has practically no organic acid level. It turns out that if we wanted to reduce the organic acid level of tomatoes, there's again no genetic variability for it. And in fact, there's nothing to even study what could possibly be the gene. So here we went to work on the melon varieties, and this is work that began with Yossi Berger, researcher in the Veillard when he did his doctorate, and was continued by Schaffer Cohen, who's now working in Ben Rimon's lab, studying genetic variability in melons, because even though I said melons, all sweet melons that we know have basically a neutral pH, there are acidic melons, and they're the facosim. Facosim, that when they're ripe, they're eaten as cucumbers before they begin to accumulate acids, but when they're ripe, they have a very high acid content, very low pH. And this is developing pH of the facos as opposed to the sweet melon. So based on those genetic studies with that material, we were able to identify the major gene that was responsible for acid accumulation in melon fruit. It was a new family of genes which actually somewhat related to the PIN transporters, proton transporter. And the mutation that occurred in the evolution of melons, which actually allowed for the evolution of sweet melons, was a 4 amino acid duplication in one of these transmembrane domains, which made the protein non-functional as a transporter. So it doesn't transport protons, acid. Turns out it's a very exciting gene because it's expressed in all fruit and is probably the major gene that's responsible for acidity in fruit. The only fruit that I'm familiar with at this point that it doesn't account for fruit acidity is the citrus, where it contributes, but it's not responsible. Anyway, so we know from isogenic melons we get this tremendous difference in acidity. We did transgenic, we silenced the gene in cucumber, and we developed cucumbers that have no acid when they're ripe, which doesn't interest anybody. And then we silenced it in tomato, and we now have tomatoes that have no acid in them. And they're completely tasteless. They're awful. Now, we did that transgenically with knockouts, with silencing, but now we did it with CRISPR. So we have CRISPR lines, which can now be used by breeding companies to develop non-acid tomatoes, which I just said doesn't taste well, but we're combining the non-acid CRISPR tomatoes with all the genes for high sugar accumulation. So we're hoping to come up with low acid, high sugar tomatoes, which will have more of a dessert taste and could be of possible interest. And I think I'm finished. So I gave you just a little overview of some of the use of wild species for sucrose hexose, transient starch accumulation. This I didn't mention, increased sink activity, Lin5, modified fructose glucose ratio, and organic acid accumulation. And we're still in the process of identifying some additional traits, additional genes and markers that can go into breeding programs to improve the taste of tomatoes. And again, thanks to all the people, Marina and Michal, who did a lot of the work, Elena, who backs up all of our work, Schaffer and Max and Arik, who were doctoral students who worked on different traits that I spoke about, Adi, who does our who works with us bioinformatically, Ran, who worked on dehydrating tomatoes, which I didn't talk about here, Shmuel Dvir and Daniel Chasson, which has now taken us into a completely different area of cannabis. All my collaborators in the Veyar, a tremendous group of people in the Veyar who really was very influential in making my last 35 years here tremendous years, and a number of people in abroad and Weitzman, and specifically to Zeraim Gedera and to Syngenta and to Breedex, who are using this material for breeding, and to the Tomazins Company and to ISF and BARD, of course, and Ministry of Agriculture, and to the Volcani. And congratulations on its 100 years. It's a wonderful place to work, as everybody's been saying. Okay.